So I've been studying coronaviruses over here for 35 years. This is how you're probably most familiar with them, people wearing masks, as is happening right now. And most of the new emerging coronaviruses that we've experienced in the 21st century are derived from bats. And in general, there are 1,300 different bat species, and each one has somewhere between 7 and 8 different coronaviruses. And so this is, in essence, a reservoir of viruses that occasionally spill over into human populations and cause big outbreaks of disease. And I'm going to try to talk to you about how this occurs, why it appears to be accelerating, and what we need to be prepared for in the future, sort of in the context of this new SARS coronavirus 2 novel coronavirus that emerged 3 months ago. So just on your right-hand side of the slide, there have been 4 major respiratory viruses that have emerged in human populations since the beginning of the 21st century, starting with SARS in 2003, the 2009 H1N1 influenza virus strain, followed by Middle Eastern respiratory coronavirus in the Middle East and Kingdom of Saudi Arabia in 2012. That outbreak is still ongoing. And then just a few months ago, the SARS coronavirus 2 strain, or 2019 novel coronavirus, or COVID-19, which is the disease. So what I'm going to talk about is talk about what the drivers are of emerging coronaviruses in terms of how they jump between species to cause outbreaks of human disease. Talk about the context of the 2003 SARS outbreak and why we were able to control that, and why this is going to be so much more difficult to control the 2020 outbreak strains. And then I'm going to talk a little bit about countermeasures that we use in our lab. So coronaviruses, nidoviruses, is a bigger, larger group of viruses that include the coronaviruses. And these viruses have a tendency to traffic easily between species. So there are 4 common contemporary human coronaviruses that we've all been infected with, probably in the first 5 years of life. They originated between 120 and about 800 years ago, mostly from bats, rodents, or cattle. To cause human outbreaks, they then colonize the human population. They cause mild disease, respiratory tract infections in children primarily. Over the last 25 to 30 years, there have been a variety of new coronaviruses that cause major outbreaks in domesticated species of economic importance, like pigs and cattle. The most notable one here was porcine epidemic diarrhea virus that killed about 12 million piglets in the US in 2012. And then since the beginning of the 21st century, there's been accelerating cross-species transmission movement, starting with SARS about 16 years ago, MERS about 7 years ago. This SARS coronavirus emerged in China 2 years ago to cause outbreaks in pigs. It's notable because it replicates efficiently in human cells of the lung, and so it has all the potential features that could make it an epidemic strain in humans. And then we have SARS coronavirus 2, which is a kissing cousin of SARS that emerged 3 months ago. There is going to be some science in this talk, but I'm going to try to keep it relatively light. What are the drivers of coronavirus evolution? Why are they moving between species? Well, these viruses have the largest RNA genome in nature. It's 30,000 nucleotides in length. And to maintain a genome of that size, they have a polymerase that has a fidelity function. In other words, it fixes errors when the virus replicates. Most RNA viruses, like flu or HIV, make a mistake every 1,000 nucleotides that they synthesize, they make a mistake. Or every 10,000 nucleotides, they may make a mistake. In a big genome like coronaviruses have, if they can't repair that, they go into what's called error catastrophe and die. So I've just told you of a potential vulnerability that if you can develop drugs to knock out that activity, the virus will go into error catastrophe and eventually burn out. So novel enzymatic functions in the virus that can be taken advantage of for therapeutic intervention. Now, this regulated fidelity can also vary depending on environmental conditions. So the virus, if it needs to increase its mutation rate to adapt to something or to a new change, it will modulate that activity. And so the viruses are quite clever. They also undergo recombination at high frequency. Well, what is RNA recombination? Well, remember that I mentioned that up to 7 to 9 coronaviruses exist in a bat. If you have a co-infection with a blue coronavirus and a green coronavirus, when the blue coronavirus is being replicated, this polymerase complex up here can fall off, bind to the green template, and make a chimeric virus. So in other words, they've genetically mixed their components so that you have something new that didn't exist before. And this happens about 25% of the progeny of a mixed infection will be recombinants. Flu does reassortment, and about 25 to 30% of the progeny from a mixed infection are resorted viruses. And this drives pandemic outbreaks of flu disease. So coronaviruses have their own trick like flu. They also can undergo modular evolution. That means genes can be shuffled, or critical virulence motifs can be shuffled between strains. Okay, so they can change quite quickly. And then if you look at the virus particle, you see these projections on the surface that give the particle a corona or a halo-like appearance in the electron microscope. That's the spike glycoprotein. It's a large glycoprotein. This is the actual structure of that protein. And it tolerates high rates of mutation, and I'll show you a picture of the amount of variation that exists within the SARS-like viruses in a little bit later. There can be big deletions. Over 100 amino acids can be removed, and the virus spike gene can tolerate it. Parts of the protein can be recombined into other coronavirus spikes. And this shuffling of sequences regulates host range, tissue tropism, transmissibility, and to some extent, virulence. So a lot of capacity to change themselves in natural situations. Now, I'm going to talk a little bit about SARS. I don't know how many are familiar with a phylogenetic tree, but this is just a way to look at the relatedness between strains. So SARS coronavirus emerged in 2003. It came from bats, and it circulated in open markets through civets as an intermediate, which were used as a food source in China. All of these strains here in yellow are strains that were associated with that 2003 outbreak. Human strains, civet strains, raccoon dog strains. That's what was circulating that was involved in infecting 8,000 people. Over the next 17 years, we sequenced coronaviruses in bats. And we found the dark red ones up there and the light red ones down here. The dark red ones are strains that are about 10% different than SARS, but can grow in human cells. They can use human receptors. And they have all the properties that would make them pre-epidemic strains, or strains that could immediately flip over and infect human populations. The light red ones down here can't, and I'll show you how that is tested. SARS2 also originated from bats, but it's nowhere on this list, on this tree. I'll show you where it is later. It's a totally different clade of SARS-related viruses from anything we had seen before in the past. And it also was associated with open markets, but that's debatable. So SARS, there are two models to explain that outbreak. The closest strain is a bat virus called WIV16, which is about 98% identical. In other words, about 600 nucleotide changes across the 30,000 nucleotide genome. It could have transmitted directly to humans in the open market, which then spread to civets and then back to humans. And as it went back and forth, you selected for strains that were about 99.5% identical that were associated with the expanding outbreak. The other model is that bats could have infected the intermediate host. Then those could have infected humans, and that set up a transmission cycle. Both are possible. What you hear the Chinese say about the SARS coronavirus 2 outbreak is that there had to be some intermediate host that transmitted to humans, and that's not actually the case. It could be direct bat to human transmission. Okay, so the SARS coronavirus outbreak went extinct around 2003, 2004. But is it extinct, or is it still present in bats? And what's the relative threat level of these bat viruses that are SARS-like? Are they dangerous to human health? And so one of the things that we do in the laboratory is called reverse genetics. So coronaviruses have an RNA genome. You can make a DNA copy of that genome and then change it however you want and then recover the virus in the laboratory. Okay? So in this case, you can remove the spike glycoprotein of the 2003 SARS strain and drop in spike genes from these other bat strains. And then ask the question, are any of these capable of replicating in human cells? And the answer is the top three, WIV1, SHCO14, these two up here, and another one called WIV16, can grow just fine on primary human airway epithelial cells. Now what these are is that transplant recipients, the discarded lung is taken. You can take the cells out of the conducting airways and grow them in the culture, so it's in essence, a replica as close as we can of what the human airway looks like. And then you can ask the question, can that virus grow in it? And these viruses replicate in those primary cells, which are shown right here, just as well as SARS can. They also use the human receptor, just like SARS, and they can use the bat and the civet receptors. So there's no reason why those strains couldn't actually spill into humans and cause an outbreak of disease. These two other strains are not viable, so they're dead. So in reality, what's happened, what the situation in nature is as follows. We have a reservoir of SARS-like bat coronaviruses that exist in bats, and this heterogeneous pool can be up to 35% different. Some of those are high risk, some of those are low risk. In 2003, SARS emerged and caused 8,000 cases and about 800 deaths with a 10% mortality rate. The prediction is that these high-risk strains could emerge in the future to cause future outbreaks. In 2019, SARS coronavirus 2 emerged, which was 22% different than SARS. It has caused 81,000 cases across the globe with 2,700 deaths in over 40 countries. There is ongoing human-to-human transmission in Germany. It's been stopped in the US, Thailand, Japan, Vietnam, China, Iran, Italy, and South Korea. So it's moving towards where we would call a definition of continuous spread on two different continents, which is the definition of a pandemic. So we're approaching that. Now keep in mind that this is 22%. There's still other strains within this pool that could emerge in the future. Even if SARS coronavirus 2 goes away, we will still be at risk for these kind of viruses. And so you need to think about it like, I know people don't do encyclopedias anymore, so I apologize. But you have to think about it like an encyclopedia of volumes that have 0% variation to about 35% variation. Some of these volumes are high risk. And they're shown there, SARS, WIV16, WIV1, SHCO14, and the 2019 novel coronavirus. They all have high-risk features. They use the same human receptor to infect the lungs. They all grow great on primary human cells. They cause ARDS, acute respiratory distress syndrome, which I'll show you a picture of what that looks like, which is what is killing people, either in humans, in the ones that are boxed, or in animals, the ones that are not. If you are older, you are at much seriously increased risk for disease. The overmortality rate for SARS and MERS was 10 and 35%, but if you're over 60, the mortality rate goes to above 50 and above 75%. And especially males are vulnerable. So I don't know whether to be glad I'm studying this or whether I feel kind of like I'm a sentinel or something. But anyway, this is the threat level. These are the strains. And one of the things that we're doing in the lab is by resurrecting these strains in our laboratory, we now have a platform to test broad-based vaccines, human monoclonal antibodies, and drugs that not only protect against the bookends, the human strains, but anything that might emerge in the future. Now, it's not just specific for SARS. If you look at MERS coronavirus that is currently causing an ongoing outbreak with 35% mortality in the Kingdom of Saudi Arabia and the Middle East, we have MERS at one end of the encyclopedia and a Chinese strain called 422, which is 35% different in its spike-glycoprotein. It can still use the MERS receptor for docking and entry. It grows fine on all the primary human cells that MERS targets. Although our intervention strategies are focused on MERS, there's another volume of high-risk strains that can cause outbreaks of disease in the future. So when you think about what's causing coronavirus emergence in human populations, the classic model for viral emergence was that you had this zoonotic pool of strains that were low risk, and you had to have a mutation occur by random chance that would allow it to either infect humans or some intermediate host. Then there would be additional rounds of mutation that would allow it to grow a little better, maybe jump to another host and then back, but successive rounds of mutation before it could actually cause an epidemic or an outbreak of disease in humans. This is rare and unlikely, so it's not a very big threat level. The paradigm, I think, that's occurring in coronavirus is that you have these generalists that exist in your animal reservoirs. These are strains that can use the ACE2 receptors from multiple species. And what's likely driving that is that as bat hibernacula is compressed, because human populations expand, you have more species of bat in the same cave with different ACE2 receptors. Selecting for generalists, you have an advantage if you can replicate in many different bat species. And by happenstance, some of those variants can now use animal ACE2 receptors or human ACE2 receptors. And so they're pre-primed, they're poised to jump between species. They may not require any mutations to drive an epidemic in human populations. They may not require an intermediate host to drive an epidemic. They may be able to traffic directly from bats to humans. Okay, so SARS coronavirus 2. It emerged in China. The first case that's been reported was December 1st. It was argued that the first cases were heavily associated with open markets in Wuhan, China. If you look at the data, the first 3 out of the 4 cases that were described had never been in open markets. If you do an analysis of genomes to ask when the last common ancestor of the human outbreak strains emerged, it would have been early November or late October that drove the outbreak. So it could be that those cases were in the open market, and that's how it actually got started. An equally plausible mechanism for the outbreak is that Chinese living in rural areas and farming communities in close proximity to bat hibernacula could have been infected with a bat. And the data that supports that is that many Chinese that do live near the bat hibernacula actually have seroconverted to these SARS-like bat coronaviruses. So they have antibodies to them because they've been exposed and infected. And if one of those individuals got infected, the incubation period is 14 days. They could have visited Wuhan, and that could have caused the outbreak. So there's a couple different hypotheses floating around that mediated the start of the outbreak, most of them, in fact, all of them going back to bats. Now, the Chinese made a classic mistake early on. They immediately said it was not SARS coronavirus, and it was a novel virus. They said it wasn't not very pathogenic, and there was no evidence of human-to-human transmission. So one important take-home message is never underestimate the epidemic potential of a newly emerged virus. They were wrong on all 3 counts. And they do have a phobia against SARS coronavirus. The economic impact of SARS on the Chinese economy was devastating. And so there's no question they were afraid that that might repeat. I would have to say that the economic consequences of SARS 2.0 has far exceeded any viral outbreak in the history of the planet as we know it right now. To stop the outbreak, they closed the fish markets, which were open markets. They didn't sell just fish. They sold a lot of wild animals. From what I understand, bats are used in certain delicacy dishes and also are used in medicinal medicines. And so there's a possibility that bat parts were in the market. But they closed the market on January 1st. They identified a SARS-like coronavirus on January 7th. It's a kissing cousin of SARS. I'll show you where it sits in a minute. They published the sequence on the 9th to 11th, allowing the rest of the world to develop diagnostic tests to begin to do rapid testing for the presence of cases in each respective nation. Around the 20th, they reported a person-to-person cluster, a super spreading event involving 15 healthcare workers. So healthcare workers were a major source of spread during the SARS epidemic. There are also major sources of spread during this epidemic. And I'll talk about the number of healthcare workers that have been affected in a little bit. So there's a lot of data here. I apologize for it, but it summarizes the major characteristics of SARS, MERS, and SARS coronavirus 2. It's a good way for you to understand why this thing is so much larger in scope than anything we've seen in the past. So let's just look at the number of countries involved. There's over 40 countries affected with SARS coronavirus 2, about 28, 29 with MERS and SARS coronavirus. The number of cases for SARS coronavirus 2 far exceeds both the SARS outbreak and the MERS outbreak. The mortality rate is about 2 to 3%, and I'm going to talk about this in a minute. There are currently about 9,000 cases that are under critical care, meaning that they're probably getting some form of respiratory support. It's about 18 to 19% of the people develop really serious disease that require respiratory support. About 83% of the cases are mild or asymptomatic. It means you can be infected with this virus and not know it. And that makes it difficult to control. The mortality rate bounces around depending on who you talk to. 2 to 3% is probably not a bad estimate, but the mortality rate in Wuhan was about 4.1%, 2.8% in a different part of the country. It's 0.17 to 1% in different areas of the world. Probably the best way to get an estimate of the mortality rate of this virus is from the Diamond Princess cruise ship, where 3,600 people were sort of locked in a closed container with a highly contagious respiratory virus that could undergo fecal spread as well. So in that case, right now the mortality rate is about 0.6%. There's about 750 people that have been infected. Most of the epidemiologists I've talked to expect somewhere around 50% of the people will have been infected in that cruise ship. About 36 are still under critical care, so whatever that number turns out to be is probably a pretty good estimate, keeping in mind that there's an age skewing toward older individuals on the cruise ship. So again, this is an example of a disease where it's good to be young and not so good to be old or male. The incubation period is 2 to 14 days. This is a real problem because now if you're quarantining people, you have to keep them in quarantine for 14 days. SARS and MERS were much more limited. The RO rate, or the number of people that get infected from one infected person, for the 1918 flu, for example, was about 2.2 cases per every flu-infected individual. SARS was about 1.8 to 2.5. MERS is low. SARS2 is around 3. That means on average, one person will infect 3 people. And this makes it also difficult to control because you have to reduce that. Asymptomatic spread never occurred with SARS. It does occur with MERS, and rarely, and that's one of the reasons why public health officials in the Kingdom of Saudi Arabia have trouble getting this thing under control because there's chains of transmission they can't track. But it's probably common with SARS-2. Why is that? We don't know. Most likely the virus is replicating in different individuals, either in the lower or the upper respiratory tract or in the nose, and this could allow for more efficient transmission. If you were a mouse, I could tell you in detail why that is working, but nobody's a mouse here, so I can't tell that. The attack rate varies depending on the situation, but 20 to 30% is probably pretty common. So those are the basic features of this. I guess there's 57 cases in the US, just to keep you updated. And I don't have the full state list shown. That's not correct. Okay, so why do people die? The major cause of death with SARS2 is from acute respiratory distress syndrome. Imagine your lung as a set of tubes that go from big tubes to smaller tubes to smaller tubes, and they end with a balloon on the end of it. And that's your alveoli. That looks like this. And normally the alveoli is open and oxygen air is flowing back and forth, and oxygen transfers across this single cell layer into a capillary bed. Imagine a capillary bed on the top of that balloon that you've just blown up. And oxygen is picked up by the red blood cells, and so that oxygenates your tissue. So what happens with SARS and MERS and SARS2 and high-path avian flu strains is these viruses kill these cells that provide the support around the balloon on the surface. And it perforates them. And so fluid pours across this capillary bed into the alveoli. And so the immediate acute cause of death is by drowning. Your lungs fill up and turn into a water balloon. Now in the repair process, hope this isn't too graphic. Might as well know. In the repair process, you end up going through what's called a profibrotic response, and you start laying down fibroblasts along this balloon to repair the perforated holes. But in some people, it doesn't just lay down a few, it continues to lay down more and more layers. And so now you have a thick layer of cells, and the oxygen can't diffuse through, and you suffocate, and you can die from this months after the virus has been cleared. So some of these patients will probably be on respiratory care for a couple of months. So this is a pretty brutal disease. 1918 flu does this, about 1 million deaths per year. The federal government has spent millions of dollars trying to figure out ways to treat this illness, not only from viral infections, but mechanical things and others. So it's just a very devastating end-stage lung disease. Okay, so why could we control SARS? Well, SARS had 3 drivers of the outbreak. The first was you had civets and raccoon dogs in the marketplace that were infected that transmitted virus to people. Well, you could take care of that by killing all the civet cats and raccoon dogs and preventing human consumption. So now the animal reservoir that spreads into humans has been taken care of. Hospitals were epicenters for expansion because of super spreaders. Examples of super spreaders: one person could walk through this room and infect 20 people. Another person infected could walk through the room and not infect anybody. But super spreaders, for whatever anatomical reason, are producing tremendous amounts of virus in a way that is readily transmissible to other people. And then you had healthcare workers who weren't wearing proper PPE to prevent infection. And so if you put in barrier nursing, you could prevent that. And the third process was that a transmission with SARS almost exclusively occurred 24 to 36 hours after infection. So you were really sick before you transmitted. And so quarantine and contact tracing worked. We don't know what the animal model is. PPE hasn't protected many Chinese healthcare workers who have been trained. 1,700 have been infected. And we have community spread and we have asymptomatic spread. So it makes it very, very difficult. If you compare the genome of SARS2 to everything else, this is the new virus. It has about 5,000 to 6,000 nucleotides different than anything else we'd ever seen. So it's completely new. Its closest relative is this bat virus called RATG13, which is about 96% identical. So it's probably a bat virus, but it's not the virus that's actually caused the outbreak. we still don't know. We still don't know what led to the seeding of that new human coronavirus. The genome of the new coronavirus looks just like SARS. Brown is replication genes. Green are structural genes. Gray are genes that regulate virulence. But there's a lot of variation in them, so we don't really know what they do. This is the receptor shown right here. And this is the part of this spike gene that binds to it. It's a perfect fit for this new human coronavirus. It's actually probably a better fit for it than SARS ever was. And so it's really efficient at using human receptors. And this is just a bunch of a little bit overwhelming data. There are 14 residues in the virus glycoprotein that bind to the ACE2 receptor. If it's green, it means it's identical. If it's light green or yellow, it means that that mutation, that change there is well tolerated. It doesn't affect the receptor interaction. If it's red, it's not good. So the high-risk strains, you see these deletions. There's all kinds of deletions in that region. That's why they can't grow and use the human receptor. But here's the 2019 strain. It's got a receptor binding domain that's very similar to SARS and some of these other bat strains, but it's also got unique mutations in it. And those unique mutations can have a big impact on a bunch of things. It can affect virus tropism, where the virus is replicating in the upper or lower respiratory tract. Virulence, although there are other genes that play an important role. Transmission frequency can vary just by changing that region. And finally, the intermediate host strains can vary. An important consideration in the US, for example, it might be that there could be reservoir species in the US that have good matches for ACE2 receptors. And so if the virus comes here, we could have an animal reservoir that the Chinese don't. And then it hangs around for a long time. So those are types of things that you need to consider. Finally, if you look at the spike glycoprotein itself, everywhere there's a green dot, that's an amino acid change. And you're going from SARS on the left side, and these are early and middle epidemic phases, to the bat strains start with WIV16. You can see there's more green dots. SHCO14 is about 10% different. This new strain is 22% different in the spike. So drugs and antibodies and vaccines that we developed in 2003 to target SARS are not going to work. We have to start over. There are some other vaccine complications that we need to think about as we develop vaccines, and there's lots of candidate vaccines that exist already. Remember that the candidate vaccines have to go through animals and then have to go into phase 1 trials humans, so we're talking months. But the vaccines for coronaviruses don't work that well in aged individuals. If you have a heterologous strain that differs by 1% to 2%, it can break through in some of these vaccines, especially in the elderly. And then you have a process called Th2 immune pathology, which basically means the vaccine primes the immune response to do bad things when you get exposed to the virus. And so the immune pathology is actually worse in a vaccinee. It's been described before with respiratory syncytial virus vaccines in the 60s, and that's why we still don't have a respiratory syncytial virus vaccine for children today. And there's evidence of enhancing antibodies, although this is more controversial. These are antibodies that don't neutralize, but they bind to the virus and allow it to infect cells it normally can't infect. Now, these are all problems with SARS. Remember, this is considerably different than SARS. They may be problems or they may not, but we have to look for sure. So that's one issue. In terms of drugs, our group formed a partnership with researchers at Vanderbilt, a fellow named Mark Dennison, Gilead Scientific, who were interested in identifying a broad-based nucleoside inhibitor for emerging viruses, and a Center for Excellence in Translational Research grant that's run out of University of Alabama to identify broad-based nucleoside inhibitors that block virus replication that would work against all coronaviruses. We have all these different coronaviruses in the lab, and so the goal was to find things that would work. And we did, a drug called remdesivir. This is an increasing amount of drug over here, and 10 to the 6 is a million viruses. As you increase drug, the viruses drop off with MERS and SARS. And just quickly to show this, it also works against all the contemporary human coronaviruses, all the zoonotic MERS-like bat coronavirus strains, all the zoonotic SARS-like strains, and all the high-risk SARS-like strains. And it also works against SARS 2.0. And it protects mice. So currently, this drug remdesivir is being used in clinical trials in China, and it's being used in Nebraska to treat US patients who were on the cruise ship that were brought to Nebraska. So it's an example of basic research that led to virus panels that could then be screened to identify broad-based drugs. The idea being if it worked against everything that you've got in your laboratory, it would work against an unknown. And finally, our collaborators. So thank you for your attention. I think I ran a little longer than I intended, but hopefully. Any questions? Yes?
The most obvious one, and one you probably hear most often, do the face masks work?
The answer is if you know how to wear them, yes. The second caveat, if you're in an outbreak setting with hundreds of patients that you're dealing with and having to wear the mask all day long, the answer is no. You're probably better off in a PAPR, which has basically a hood over your head with a respiratory filtered pump on the back that pumps air up through the hood down across your face to give a curtain and air. So I would say if you, in a scenario with a full-blown outbreak with large numbers of cases and a lot of virus in a hospital setting, you're probably better off with more intense personal protective gear. Yes?
You just said it works if you know how to wear it. How do you wear it?
So the first thing you should do is go to the CDC website. They have a detailed description of how to don and doff the mask. A lot of people make a mistake when they take it off. So I would go to that website and take a look at that. Yes?
Does it pay to get a mask that's not an N95 mask? You might want to explain what that is.
So an N95 is a mask that removes 95% of the particulate matter in air as you breathe in. So in a research setting, the minimal respiratory protection we can use working with SARS or MERS or the new coronavirus is an N95 mask. My lab doesn't like to do that because it's the minimal requirement, the minimal protective amount. I would rather be at a level higher so that your mucous membranes are protected and you have additional barriers that protect. That's not available for the public. So the public is going to be a surgical mask or an N95 mask. There's no question the N95 mask would work better if you wear it correctly. The problem with that, especially if you're older, is that it causes respiratory stress. It's hard to wear a long time and still breathe easily. And so there's a debate among public health practitioners whether the N95 is better than just a straight surgical mask. Also depending on age, health status, your respiratory function, those are all factors that make a difference. Yes?
Can we read anything into the fact that the cases reported so far seem to be all in the northern hemisphere? Does that have anything to do with the virus itself?
Well, except for Australia.
Oh,
Right. Australia has a fair number of cases. I would probably argue that the bigger reason we haven't seen any cases in the southern hemisphere is that diagnostic testing is not being done, sadly enough. I think there are 2 places in Africa, for example, that can actually do diagnostic testing. There's not enough diagnostic tests available in the US right now to do the kind of screening we might like to do. Hopefully that will change in the near future. But I would argue it's more likely a factor of not testing. Yes, sorry.
So you went over differences in terminologies, which were really helpful, but I'm still kind of confused. obviously there are so many different terminologies. You know, like the virus itself is like COVID, not COVID-19, like the disease itself that like originated from the virus is. So a lot of the health organizations still call coronavirus as a novel form of virus, and I think at one point you did mention that because it, you know, it's like it originated from like the SARS-CoV-2. Like, is that still like what are some like accurate terminology to refer that to better really?
So the global coronavirus taxonomy group has said SARS coronavirus 2 is the appropriate name for the virus, and that COVID-19 is what the WHO calls for the name of the disease, not the virus. There's a lot of people who aren't happy with SARS-2. For example, in the US, SARS from 2003 is a select agent. So now in my lab I have SARS and SARS-2. This could be a problem, just in terms of keeping vials separate. So we're actually naming things in the lab different so that there's a clear designation separation between SARS and the new coronavirus. But in general, the coronavirus taxonomy group who has the authority to make the call has said it's SARS 2.0. There's lots of scientists that aren't happy with it, but it does reflect the disease, which is very similar between SARS and SARS 2.0. Severe cases are developing ARDS without bacterial secondary infections, very similar. The main difference is the transmissibility and the large number of mild and asymptomatic cases that occur with the new strain. Okay, I hope I answered your question.
Thank you.
Okay. Yes, sorry.
You had mentioned that 80% of cases are mild or asymptomatic. Would those typically require hospital stay, or whether?
They typically don't. They may, oh, I take that back. Some go to the hospital and don't require respiratory support. So the correct answer is that of that 83%, there are some that are in the hospital and some that aren't. And I can't tell you the fractions. The data is too murky in China. But there's no question that there are cases that go to the hospital that don't require respiratory support. About 17% are serious or critical, and they do. Yes, I think you had a question.
Yes, the CDC's press release yesterday has gotten a lot of headlines. I'm just curious your thoughts on whether that's an accurate reflection of the scientific communities, where their heads are at the moment.
This is the sort of the forecasting of what's going to come. So every modeler that I have talked to who models infectious diseases says that the new virus is coming to the United States. I don't think we can escape it. Even extremely aggressive quarantine tactics have trouble stopping a virus that can undergo asymptomatic spread. However, those kind of tactics do delay and reduce the rate of spread. There is an advantage as I have to be careful about this. There's no question that respiratory viruses transmit in the summer. But their peak transmission time with the highest RO is in the winter. So as you move into the summer, the virus transmission rate to the number of people that you can transmit has a tendency to drop some, and now it's more vulnerable to public health intervention strategies. So really aggressive public health intervention strategies as you move into the summer could actually stop it. But in general, we could quarantine China, but are we going to quarantine Italy? And if cases spread to France, South Korea, Singapore, Indonesia, how many countries can you quarantine before you are basically stopping your own economic production? So that becomes this very delicate balancing act, which is impossible to predict, in my opinion. So, unless you do machine learning, and then maybe you could predict it. I don't know. Yes?
So you talked a little bit about this, I think, but what do you think the likelihood of it becoming like a seasonal disease? Like people have talked about it becoming like the flu, where it's here just every single year, and we have to deal with it from now on.
So my prediction would be that it would act like a contemporary human coronavirus. So the danger of new emerging coronaviruses is that everyone is naive, especially adults, and the virus has a tendency to cause more serious disease as a function of age. So for example, if you're under 15, as far as I know, there's only been one death. There have been lots of kids that have been infected, but most of those are mild or asymptomatic. They're closing schools to prevent transmission in a school setting that would then spread to adults. Once the virus colonizes the human population after the second year, first 2 years will probably be pretty brutal. Attack rates of 30%, mortality rate somewhere between 0.2 and 2%. That's going to be pretty brutal, but eventually most of the adults will be infected so that the virus will now be infecting children where it's mild, and now they're immune for life. SARS2 is a mild disease, it causes common cold, just like every other coronavirus. So I don't think it would be like flu. I think it would look like one of the other contemporary human coronaviruses. Once you get through that first couple of years of colonization where the elderly are most at risk. Yes?
So, going back, you're talking about, so the R rate, the R naught, you're saying is 3, but just how bad do you think this could get? And like, what would it become a pandemic? I know currently right now the Chinese government is talking about reinstating eventually economic activity, you have supply chain. From a CDC perspective or public health, would that be a smart idea? And if they were to reintroduce or restart activity, that would allow for potential increases in transmission, correct?
So the purpose of quarantine is to limit transmission chains by locking people up in their homes. And so they don't come in contact, and so the virus has a tendency to burn out because it doesn't have enough naive cases to maintain itself in the population. No one's ever tried to quarantine at the level of 100 million people like the Chinese did, so that was quite novel. I'm not one, I don't know of anyone in the West that really knows how well that worked. I mean, it seems to have worked somewhat, but that means that you have a fairly large naive group that has stayed home. When they go back to work, asymptomatic spread would repopulate those individuals and you'll have another wave of disease, most likely. I think that's what the modelers would probably tell you. How bad can it get? I could do the math for you if you want. I don't think you want me to do the math. I don't, let me see. Okay.
So you remember Michael Crichton's Andromeda Strain?
The what?
The Andromeda Strain, you remember that?
I saw that movie.
Of course you did. Of course you did. You're not supposed to admit it though, right? You're too young for that.
Well, I'll take that. I'll take that compliment. It's not true, but I'll take the compliment, yes.
So what about vaccine? Obviously, that's the V word that's out there. What about opportunities for labs, private or government funded? As an entrepreneur, if you could put that mindset together, what would you be doing right now to try to take advantage of this opportunity?
So I'm one of those silly academicians, right, who worked with a company that had the IP control of their drugs to identify which drug would work the best. And so I was happy with that. We obviously, as a research group, have a fair number of patents, some of which may be worth something, some of them may not. I personally think that even if SARS coronavirus 2 goes away, let's say it goes away, the fundamental ecology of these viruses are going to argue that it's going to happen again and again and again, and one of these is going to be just right, with the right amount of mortality, the right amount of transmissibility, that it can't be stopped. And so I think as an entrepreneur, I can't say that word, our group is working on broad-based vaccines for SARS, and the SARS group of viruses that I showed you and the MERS group of viruses that I showed you, so that we would have vaccines on a shelf. As an academician, will I make money from that? Most likely not, because you're preparing a product for something that hasn't happened, and you don't really know how soon it will happen. So the only real purchase group that would purchase that would be government agencies that would want things on a shelf. So I think I made that decision a long time ago to stay in academics and not into the corporate world.
But as an advisor, how would you?
I tell my students to go work in the companies. Yes.
You talked about respiratory and fecal spread. What about surfaces, hand-to-hand, other types of transmission?
So the one report that I've seen so far argues that depending on the matrix, so if you purify virus and put it on a surface, it doesn't last that long. But if it's in fecal material or respiratory secretions, it can last 5 to 9 days on the surface. So that's probably what's contributed to that, and potential for fecal oral or fecal respiratory spread probably has contributed to a fair amount of the infections on the Princess cruise ship. They weren't expecting that kind of transmission and fomite transmission on surfaces. And so that probably is a bigger problem than we thought. During the SARS outbreak, the Amory Gardens was in Hong Kong, and the toilets were designed so when they were flushed, it caused an aerosol plume that would go into the air handling system. And so most of the cases in Hong Kong were because they were breathing in aerosolized feces with high concentrations of virus. And there was so much virus that the plumes that came out of the building drifted over the next big apartment building and infected people in there too. So that's not the normal route of spread, but you might imagine any kind of toiletry that like on an airplane that can cause a vacuum or aerosol spread could contribute to spread. Am I making anybody more or less comfortable? I don't know. Yes?
You mentioned something about a treatment that seems to be working. Can you talk more about that?
Well, I don't know if it's working or not. All I know is that they have set up cohorts and that they're testing the drugs. So remdesivir also had broad activity against Ebola, and so it was tested in Africa. It did not work as well as therapeutic human monoclonals, but it did reduce the mortality rate from about 70% to about 50%. The key for any of these treatments for this disease is early diagnosis and early administration of drug. Because ARDS, as I mentioned, is an immunopathologic disease. So not only does the virus cause damage, but your immune response causes damage. So at some point, the virus titers drop off, and it's all immune-based. And so if you drop your antiviral in too late, it has no effect. So the same with Ebola. So you have to get these drugs in as early as possible to maximize the effect. And it works against these SARS group 2B coronaviruses, not only in animals, but mice and other species, but also primates. So they should be testing it. Whether it'll work or not, I don't know. Yes?
Can you mention the drug name one more time?
Huh?
Did you say the drug name?
Oh, it's called remdesivir, or GS5734. It's a Gilead compound.
How available is it?
Huh?
How available do you expect it to be?
They certainly made enough to provide the Chinese for 2 large cohort studies, and they have made enough for the U.S. government. I think well, I don't want to speak for the company. I'd probably get in trouble. So, but you could talk to the company. They're certainly very interested in improving the bioavailability of this drug. For example, right now it has to be given IV. They are interested in tablet form. Yes.
So if you're asymptomatic, you're a carrier in essence.
In essence, you are. Yeah. Your main job is to transmit the virus to those who haven't been infected. In terms of what the virus is interested in you for. Yeah. Yes.
Out of curiosity, has there been such a thing as an asymptomatic super spreader yet?
That's a really good question.
And
I'm 90% certain the answer to that is yes, but I might be wrong. And if I am, I apologize to the group. Yes?
I don't believe everything I read, but I did read that someone had been exposed and was not symptomatic for 27 days. Is that possible?
I've heard that report too. That was someone in China, wasn't it?
I don't remember, I'm sorry.
At this point, I can't give it any credibility. The important thing to remember is that we are outbred, right? Human beings are outbred. Each of us are genetically very heterogeneous. And so how we respond to infection might be very different. So the question you really asked is, are there rare outliers out there that may have a different transmission window than what 99.999% of the population has? And the answer to that question is probably yes. Yes?
I also read that there was malfunctions in the testing kits that are in the United States. Is that true?
Yes, that was true. The CDC had to bring them, have them sent back, and then they are redistributing them.
How are they malfunctioning?
You should check with the CDC. I think I know the answer to this question, but I'm not 100% sure, and I don't want to be wrong. But there is a problem with the kit, so they're redistributing it to public health agencies. Yes?
What is the unique furin coronavirus as opposed to like influenza or pneumonia that has high death rates as well?
How many in the room have been infected with influenza?
Ever?
All of us. Everybody's hand needs to go up. How many have been infected with SARS2? No, fortunately, it better be the case. None of us have. So number one, it's scary. Number two, the mortality rate for flu, contemporary flu, is usually less than 0.1%. So 35,000 to 70,000 US citizens will die each year, depending on whether it's a severe strain or not. This coronavirus has a potential mortality rate of 2%, attack rate of 30%. We have 300 million people in the country. If you do the math, it's scary. Okay? So it's scary. And it's a false equivalency to say that we need to be more worried about flu. We need to be worried about flu. It is, I would agree, that it's probably the most dangerous virus on the planet as a respiratory virus that can cause massive human morbidity and mortality. But any respiratory virus that is easily spread and has a potential mortality rate of up to 2% should be taken with a tremendous amount of seriousness. And I don't think it's in a public health official should not be saying that we need to be more worried about flu.
No,
Yeah.
I wasn't making that.
No, I know. I got up on his box, didn't I? I was lecturing. I'm sorry. I didn't mean to do that. But I was actually lecturing other people that weren't in the room. Yes?
So we see most of the research that's been focused on the spike protein. Are there any other targets that research groups are working on?
So the nucleoside inhibitors like remdesivir target a protein called NSP12. It's the RNA-dependent RNA polymerase. There are small molecule inhibitors that can block other replicase proteins like the proteases. So there's a big genome at one end of the virus, and it encodes 16 proteins that have to be cut into individual proteins by a viral enzyme. And that's a really good target for antivirals. And there have been a number that have been developed against SARS. Because SARS went away so fast, their development lagged. Companies didn't become interested. Funding agencies weren't getting enough impact to support it, so they weren't getting funded. And so a lot of those compounds just ended up in somebody's freezer that are now being rapidly pulled out and retested against the new virus. So there's a number of other of novel targets that could be attacked with coronaviruses. And to some extent, it's got a very unique replication strategy And it has about 5 or 6 novel enzymatic activities that aren't present in any other RNA virus that can be targeted. And so it actually has more vulnerabilities in the genome than like flu might have. And so with effort, I think we could have a large number of good compounds that would work against the virus.
Just one more follow-up. How long do you think that it's going to take to get from the clinical one trials to actually have the product out in the field?
A vaccine or a drug?
Both,
Small molecule inhibitors can be used under compassionate use if they've been tested in 2 animal models. And so those can reach the clinic fairly quickly in an outbreak setting where you have a fairly high morbidity and mortality rate. And so that can occur quickly. And in fact, the drugs that are remdesivir is being used under compassionate use right now. The Chinese have tested a large number of different things. I would imagine that they already have therapeutic human monoclonals in clinical trials there. They had about a month and a half head start over the US. The US should have therapeutic human monoclonals within about a month, and those are going to have to go through toxicity testing and tissue specificity testing and potency and stability, and those are 4 to 6 months away. Okay? Yes?
One more question. So for those who are traveling, how should they be worried about catching coronavirus on a plane?
Yeah, I worried about that this morning coming over here. I had to review grants for NIH last week. I had to fly to San Francisco, and I'm thinking, what am I doing? I would certainly carry a mask with you. Whether you want to wear it or not is a fair question. I did not, because I thought it was a little bit early. But I pay attention to where cases are occurring all the time. And I guess the big question in the US is that are there asymptomatic transmission networks that are occurring, that have occurred and are currently expanding that we don't know about? Right now, it's a really bad flu season. And so the tendency is to say, well, it's probably flu. And if we don't have enough diagnostic kits, we won't know it's the novel coronavirus. So I would say that, we're a little more vulnerable in the US than we would like to be. And until we get those sort of a standardized, highly sensitive PCR-based kit or serologic-based assays out on the market so that in large numbers, I don't know how much, we really won't know what's happening in our country. Yes, sorry.
The Chinese government said it was like a strand of SARS and not a novel virus. Would the numbers look a little different today?
Boy, that's a hard question to answer. They certainly convinced their public health officials in Wuhan and in the province that it wasn't virulent and not very transmissible. And so they had large government celebrations with literally tens of thousands of people in close proximity in Wuhan in early January. That this is what you don't do, is you bring large numbers of people together in confined spaces is the perfect environment for respiratory virus spread. So I would say because they downplayed the seriousness of it, they probably caused the outbreak to be much worse in and around Wuhan. And once that became clear that it was out of control, the rest of China responded quite vigorously in terms of public health intervention, quarantine, restriction of movement, contact tracing, and so on and so forth. So the number of cases in Beijing were much less, and other cities were much less. So, yeah, never underestimate it. It's a lesson for us as well. Yes?
Do you have specific actions you'd like to see from Congress in the short term and longer term?
Well, I think the plan to put money into the the efforts to contain the spread of the virus is a good plan that the president put forward. Long term, in the short term, we need to develop a vaccine against the SARS2 strain, and we need to identify better small molecule therapeutics that are like orally bioavailable. In the long term, the problem is clear and it's not going to go away, so we need to think about funding, government funding for broad-based vaccines and broad-based drugs that can be put on a shelf so that we're not so vulnerable. In the case of SARS, the epidemic came and went very quickly, and so there was never a vaccine, and then all efforts stopped. 17 years later, it's unfortunate that all efforts stopped. And we should learn from that. 3 new coronaviruses in 17 years is something that you should pay attention to. So thinking about putting it at a higher priority for, not necessarily for funding, but also for developing public health intervention and therapeutic approaches would be a good plan for the future. I don't generally do policy, so.
Thank you so very much.
Thank you.